Hello, everybody. I'd like to tell you a little bit about the Shusta Research Group. So first of all, I'm Eric Shusta. I'm the leader of the Shusta Laboratory. Just as some potentially interesting facts, I grew up here in Wisconsin in a suburb of Milwaukee called West Allis. I did my bachelor's degree here at the University of Wisconsin-Madison, followed that with a PhD at the University of Illinois, and then a postdoc in neuroscience at UCLA. And what really inspired me to pursue graduate school and then eventually a faculty career was that I was interested in cutting edge research, trying to explore the unknown. And what research are we interested in the Shusta Laboratory? We really use a lot of different molecular and cellular biotechnology tools to look at problems of drug delivery, in particular those problems related to delivery of drugs through the very impermeable vasculature of the brain, known as the blood-brain barrier. What do I do for fun? Well, in Madison, as you know, we are in an isthmus between two lakes, Lake Mendota and Lake Monona. So I like to take advantage of the water. And so I just have a nice picture here of Lake Monona and what it looks like at sunset. And what do I like most about UW-Madison? It's really a collaborative, collegial environment, top programs throughout the campus. And as you'll see through my short vignette today, we try to leverage that in our research. So here's the team. I'll let you look at this on your own. We have a nice group of students comprising both postdocs and graduate students from different backgrounds. And so these are really the people that enable the cool cutting edge research that our laboratory does. So as I mentioned, we use these molecular and cellular biotechnology tools to address this problem of drug delivery to the brain through the impermeable vasculature. This is one vessel that I'm showing here. A drug needs to get from inside this vessel into the brain tissue. And because of the tight junctions of the endothelial cells in the vasculature, it's very difficult to deliver drugs into brain. So we use two parallel toolkits. One is with protein engineering. We like to discover antibodies that can transport across this cellular monolayer, evaluate pharmacokinetic and biodistribution of these antibodies within the organism and in vitro models. We like to conjugate these then to drug-containing particles for therapeutic development. In a parallel arm, we work with induced pluripotent stem cell technology, a lot of times in collaboration with Dr. Sean Polachek's lab, where we try to come up with new and improved in vitro models of the blood-brain barrier so we can understand what the blood-brain barrier looks like in disease, and then we can leverage these models again for therapeutic development. So I'll just give you a couple minutes on a recent paper from the lab to give you an idea of the types of things we work on. This was a recent publication in Science Advances with Ben Umlauf, a former postdoc in the lab, as a principal author in this study. And the idea is that in humans, primary brain cancer, an advanced form of that called glioblastoma, shown here in this cartoon, are very debilitating cancers and have very short lifetimes and survival rates, despite the fact we're doing our best in terms of supplying medicine. So usually on the order of 14 to 18 months survival rate. So in our lab, what Ben and others have done is try to bring a new approach. The idea here is that in an intact blood-brain barrier or an intact brain vessel, material does not move from inside the blood into the brain tissue. However, in tumors, there's some leakiness. So perhaps if we have the right targeting agents that can get into these leaky areas and bind and accumulate selectively in the brain tumor region, we could now have a new way of targeting the pathology in order to deliver therapeutic solutions. So we're also developing new molecules to act as these targeting reagents. We use something called a VLR. This is a binding protein from lamprey that's very nicely designed to bind targets in this concave cleft. So what we then looked at with these particular molecules that could target these openings in the blood-brain barrier selectively in the GBM was to use mouse models of human brain tumors and look to say, where do these targeting molecules accumulate? And it turns out that in green here is the targeting molecule. You're looking at sections of brain tissue. In purple is the vessels. In green is the binding of our targeting reagent. You'll see in the tumor, we see very nice green from this P1C10 molecule. In normal brain, we don't see any uptake. So if we're targeting a chemotherapeutic, it will only be toxic then to where it accumulates in the brain tumor. And we also don't see this in the peripheral organs like kidney, heart, and liver. So we were very excited about this specific targeting. And then that translated into new survival metrics for us in this animal model. You can see in the red curve here, this is with the P1C10 targeted liposome loaded with chemotherapeutic, compared to untargeted or mock targeted liposomes also loaded with the same chemotherapeutic. You can see this shift in survival time of about 2 weeks, really indicating that targeting matters and that these approaches that we're using may offer us in the future new approaches to targeting brain cancer. So with that, I'll just wrap up by saying there are some exciting parts of this story. This was really mediated by a graduate student and a postdoctoral fellow in the laboratory. These are two great researchers. Ben Umlauf is now an assistant professor at the University of Texas at Austin. Jason LaJoy took advantage of NIH-funded fellowships here on campus in the biotechnology program and the genomic science training program. And he's now at a startup company doing antibody development. I talked about the collegial environment up front. So Dr. John Kuo and Dr. Mahua Day, two neurosurgeons and brain cancer treatment experts, are really collaborating with us both on the initial study and moving this forward into new immunotherapy paradigms. I should point out that people who work on this within the lab are part of patent applications. They're part of those invention disclosures. And that has even led, in this case, to an industrial collaboration with Sigilon Therapeutics, where they're testing out some of these molecules for their properties using some of their proprietary systems and therapeutic cargoes. And of course, all of this comes from funding. So we get funding from a diverse range of places, including award type funding and also the National Institutes of Health. So with that, thank you very much. And I'd be happy to answer any questions you might have or interest in the laboratory. Feel free to reach out and email me or check us out at the lab website. Thank you very much.